Okay, got my bread from our baker in Waco Station, and now I'm on my way to my lab. Hi! Ohio! That's not good. Let's go. Okay, now I've arrived at the RIKEN Waco campus, and this is actually one of the biggest campus for all RIKEN campuses around Japan. And this is our central brain science building, and we are at the deeper end of the campus, but it's about a 10-minute walk from the entrance. And now I'm heading into the building. Our lab is located on the 5th floor, so after this, on the elevator. So we get off here, and now this is our lab. We are the lab of neurodiversity, and we study how neurons develop in flies and mammalian cells. And it's Jason. Hi! Alright, I'm gonna start my experiment. I'm gonna be using these larvae today. They look good. Alright, so I'm gonna take them out. So here I've taken them out in this dish. I'm gonna look them under the microscope and check to see to how their neurons look like. So I'll be using this machine. Here's my sample. I'm gonna set them here, and that's how a neuron in a larvae looks like. Now I'm gonna take images of these and plug them into this program to find out how many number of ends they have. Okay, so I'm back in the fly room, and I want to see if anyone's using the microscope. It's brand new. Okay, Funky's using it. I wonder what she's doing.
Oh, today I'm going to check the neurons expressing the neuronal activity indicators under the scope. And with this imaging system, I can try to activate the neurons with different methods and observe how they react to different stimulus. And our lab is also interested in studying how different genes are involved in building the neurons and how the behavioral outcomes are affected.
It's pizza! So for my next experiment, I need to use chloroform, which I can't find. So I'm gonna go look in this room. Oh, hi Jason. Do you know where my chloroform is?
Hey, I think Adrian has it in his office.
Okay, I'll go ask him later. By the way, what are you doing today?
Purifying DNA. We're going to use this DNA to make like transgenic flies, and we're going to study neural morphology in the transgenic flies.
Ooh, okay. Alright, cool. Alright, I'm gonna go look for my chloroform. So thanks. I'll bother you later.
Yep, see you.
Okay, bye. Alright, so still looking for it. I can't find it. Anyway, so this is our lab, and we have a couple of benches. We have a lot of equipments too, but the chloroform is still missing. So I'm gonna go into this other room to check it out. So what are you culturing?
So we are culturing several different types of cells for our cell cycle related experiments.
Okay.
So we are going to conduct some live imaging with the fully positive cells to look at the cell cycle dynamics under certain conditions. So currently our new student,
Okay. Hi.
is working with these cells, and from next week on we are going to set up some experiments to image them with our new microscope.
Awesome. Hi.
Oh, hi.
Hi. I want to talk to you a little bit about our lab today. And if you could show us what's great and what we're doing, that would be fantastic.
Okay, yes. I'd
All
like
right.
to introduce everybody to the research we're doing.
Okay, thank you. All right.
Okay.
Hello, I'm Adrian Moore. I'm the PI for the
Laboratory for Neurodiversity. We're a group of neuroscientists, geneticists, cell biologists, and people interested in imaging who are studying the process of neural differentiation. We're interested in what happens from the early birth of a neuron through the formation of all its differentiation steps to its integration into the mature circuit. We know the formation of this complex, branched and interconnected structure is critical to the formation of circuits and their function, and it's disrupted in neuropsychiatric diseases, and it's failed to be maintained in neurodegenerative diseases. So we're interested in how this occurs, how the differences between different neurons different neuron differentiation programs are programmed and read out during the process of neural differentiation, how it's coordinated across multiple cells at a systems level, and indeed how it can be reawoken and reutilized, perhaps even modified and programmed to aid with neural regeneration. So within the lab, we use multiple different techniques, genetics and genome engineering. We use a lot of profiling techniques. We use in vivo imaging techniques, as I'll show in a moment, and we use circuit function, looking at circuit functionality and behavioral activity. So one of the things we're interested in, how neurons select and guide their programs. Well, ourselves and others over the last few years have shown that this is through transcription factor codes in the newly born neuron. And for example, several years ago, we showed this in the neurons which are mediating this escape response. These are nociceptive neurons just under the surface of the cuticle of the Drosophila larvae. And we showed a transcription factor code which functions in class 4 neurons. And if we manipulate that code in the newly born neuron, and this was done by Shiho in the lab, we can get the class 4 neuron, which had been fated to become a class 4 neuron, to switch its differentiation program, in this case, to a second neuron type, the class 3 neuron. Moreover, if you're interested in understanding how the process of neural differentiation, you really need to be able to image all those steps of dendrite arbor formation in vivo over time. And this had proven very difficult to do, but Li Feng in the lab worked out that if she used the Drosophila pupae, because the pupae don't move, if she could find neurons that are undergoing these differentiations, she'd be able to follow those through all the different stages of arborization, from initial outgrowth through to final complex arbor formation. So she did this by following arbor formation for over 70 hours in the developing surface sensory neurons in the Drosophila pupae. And then once she'd worked out this process, she decided to go back and look in detail at one particular process that she saw going on. And that is during the early formation of the arbor, where she saw that this tip would undergo pre-programmed splitting or division into major branches that were left in the wake as the tip grew out. So what she did was she then went and looked at this process by zooming in on the tip of the dendrite as it undergoes this branching. Here she's used DFP to mark the tips of growing microtubules, and here she's used red fluorescent protein to mark bundled actin. Now, if you put these two together, you can see that the microtubules the actin is growing in from the periphery towards the center of this dendritic tip growth cone and acting as a site to guide repetitive microtubule targeting into the periphery. When this structure resolves, you see that microtubules have been targeted to form the new branches. So how does this process function? Well, I don't have time to go into detail, so please go and look at Li Feng's paper. But what she did is she then took a genetics approach in Drosophila, taking advantage of the fact that we know that there's disrupted arbors in these diseases, and looking at families associated with predisposition loci, screening these in vivo by knocking them down in the neurons and asking how the development of the neurons is disrupted, and then going ahead and digitizing these images to allow us to quantify different aspects of arborization over time. Therefore, being able to draw ethograms to look at the behavior of the neurons over time and also to quantify that. So that was done by Li Feng with help from a student in the lab, Wei Kim, who was a student here and also registered at University of Science Malaysia. And if you're interested more in what I've talked about, Yunjin in the lab recently published a review on the transcription factor guiding of arbor formation, and Ollie, a student in the lab who just handed in his thesis last week, published a review on the microtubule control of arborization. So thank you very much.